thank you very much for coming to my talk, which is titled Does Size Matter? Giant Sperm and Caenorhabditis Worms. And this is work that I'm working on together with my PhD advisor, Dr. Asher. Since we are in a speciation section, I don't think I need to convince anybody that the diversity of life is amazing and super interesting in itself, but I am going to try and convince you that the diversity in sperm is also really interesting and should be studied. These are a variety of different types of species from all kinds of, basically anywhere within the animal kingdom. Lots of them are diverged into like all kinds of strategies, basically. Essentially, sperm are the most diverse types of cells. They are also the only types of cells that are basically released into the environment to go do their own thing. No other cell type really does that. I suppose pollen, but that's the same thing. So they're really, really cool. But for some reason, evolutionary biologists didn't really take too much of an interest in them until Parker, when they realized maybe sperm aren't just incredibly diverse because the sites of fertilization are very diverse, which could be external, internal, female body, could be in the deep sea or anywhere else. There must be other reasons why we see the diversity that we see. And some of the diversity could be a result of sperm competition, which is obviously really interesting to an evolutionary biologist. And one of the probably most popular examples is the fact that sperm might be really, really big, even though theory predicts it should be numerous. And as a result of being as numerous as possible, it would be small. But then sometimes it's really huge. As you can see here in the Drosophila example, where this is the sperm and this is the male. But that's also something that we can see in mammals, birds, fish, amphibians, and as I said, insects. However, I study giant sperm and Caenorhabditis, so they also have that. The most popular Caenorhabditis, I suppose, is elegans. And you have a tiny scale here that measures spermatid size. This is how big elegant sperm is. But then across the genus, there's really huge variation in how big sperm can be. And what that looks like under the microscope, here you have elegant sperm, the standard kind, and then you have macrosperma, which is really big sperm, and plicata, with the biggest type of sperm. So they're really big. And I need you to forget that sperm usually has a head and a tail, because worm sperm is amoeboid. So this is what it looks like. It just crawls around like little amoeba. So that's basically what I'm studying. I have a favorite species pair that's going to be the majority of what I'll be talking about today. They are these two, macrosperma, conveniently named in a way that you will be able to remember it has the big sperm. And I'll use this image to refer to a big sperm species, and then Noragensis, which is very closely related and has small sperm. They've both been collected from French Guiana. So I'm studying this in Caenorhabditis, which are amazing model systems to study sperm because they're tiny, but they're also transparent, so you can actually see the sperm in action. And they're very promiscuous, which is very helpful, and they mate non-assortatively, which means I can mate species that don't actually mate with, they don't produce fertile offspring together. That's all really helpful. Okay, but my questions are, why did larger sperm evolve in Caenorhabditis? And I've already hinted at the fact that it might be due to particularly strong sperm competition in some species. And it has been shown within species that larger sperm means faster sperm and better at displacing other sperm. So it could be that these species that have giant sperm just really, really do have particularly strong sperm competition due to life history. Okay, so first off, I wanted to look at some general traits related to sperm that are basically relevant within species, such as whether the size of sperm somehow affects the number of sperm that are being transferred in a single mating, and whether that in turn affects lifetime fecundity. So that's what I'm going to show you here. This first data set is mine, that's from collaborators in Nice. And basically they're comparing two different species pairs, one with small sperm and one with big sperm. And every single time they show that bigger sperm species transfer fewer sperm in a single mating. So then I was wondering, does that mean that they have lower lifetime fecundity? And this is the data set for females, for macrosperma and Noragensis. And in fact, macrosperma do make fewer offspring in the females in their lifetime. But then we can see the same thing in males, where macrosperma, which is here, has fewer offspring over their lifetime than Noragensis does, which is the closely related species with small sperm. Next, I was looking at differences in what happens when these species mate with each other, which, because they're non-associative maters, can happen quite frequently in nature. And yeah, I was basically wondering, does that make any difference? But first of all, maybe it's a summary. What happens when these species interact in accidental interspecies meetings? So that question didn't come out of nowhere. That's basically based on something that had been found in our lab by Jen Sting previously, which is that even normal-sized sperm, sometimes when mated with the wrong species, can harm the females. And what you can see here is the head and the center of the body with the uterus being in this area of a female, and all these glowing little dots are sperm, and they're clearly outside the area where they're supposed to be, which is just this yellow bit and then the blue bit in this area, but they got all kinds of places outside in the body, which is where they really don't belong. And this effect has been correlated to a reduced lifespan and reduced fecundity. Okay, so this is basically what it's supposed to look like when mating goes well and sperm just goes places that it's supposed to be, and then as a reminder, this is what it looks like when it goes places it should not be. So I was wondering, if giant sperm are more competitive, does that mean that they're also more harmful when they mate with the wrong? If they're better at displacing, maybe they're better at mating. So that's basically what I tested. Just a reminder, I'm looking at Noragensis and macrosperma. And just so you get a feeling of what I'm talking about, if worms were the size of people, Noragensis would have sperm that's about this big. Macrosperma would have sperm that's about this big. So when I talk about invasive sperm, imagine this in your body, or you don't. Anyways, because you can't really manipulate sperm size in worms, what I did was compare these two species to a third species, which is elegans, in a common garden type approach. And I did this by scoring whether sperm is mislocalized or not, and in how many females that's the case. I already mentioned they have see-through bodies. We can stain the sperm so that it glows. It's amazing because we can look at sperm in vivo. And basically I score 4 different locations, which are here, the uterus and the spermatheca, which is the site of fertilization, and then the gonads and soma, which are both places that it should not go. So I'll dive into a bunch of results. They're all going to be the same type of graph, so I only walk you through the axis the first time, but it's going to be pretty much the same for all data sets. Okay, so what you're looking at is the proportion of females that had some kind of invasive sperm, so in the gonads or in the soma. And then down here is macrosperma and Noragensis. So when mated to an elegans female, Noragensis sperm invades in approximately 20% of the cases, macrosperma in 90%. So the next question was, what happens if I take these two species and I mate them to each other at the reverse and compare that to their normal within species matings? So I did that with Noragensis. You have the small sperm species mated to each other, basically no invasion. The small sperm and the big sperm females, essentially no invasion. But then the big sperm and the small sperm species females, 70% invasion. But also the big sperm species within its own females has 20% invasion. So they're clearly doing this, not to the same level, but even to their own females. And here's what that looks like. So you have a worm, head and tail, and here's where the uterus is. And then, I don't know if it's really visible with the light being this bright, but basically sperm goes all the way into the head. This kind. The next step was to compare 2 different genotypes of macrosperma that we fortunately have. They have slightly different sperm size. They're both on the bigger end of the scale, but they're slightly different in size. And I compared them to the small sperm species, Noragensis. So you can see here, they measured the spermatid size and they differ a little bit. So in these 2 different genotypes of the same big sperm species, the bigger one invades in 85% of the cases, and the smaller one a little bit less. So it seems to be actually correlated to the size. Then we were going to look at whether it does this, whether they're just generally more invasive, or whether they're also good at doing this really, really quickly, because maybe squeezing through the tissue kind of paves the way for more sperm to come after. So this was the first step in looking at that. And as you can see here, this is the smaller of the two big sperm species, and then this is the larger one. And it doesn't only invade more, it also does this earlier on. This is all on time scale from ½ an hour to 6 hours, by the way. Okay, so then we wanted to compare whether females just differ a lot in their susceptibility. And there's the small sperm species Caenorhabditis that we know has very, very invasive sperm and tends to invade pretty much all females it's being mated to. And we use that as a positive control for invasive sperm in macrosperma females and Norwegensis females. And when mated to Caenorhabditis, the small sperm species is basically invaded in 100% of the time, whereas the macrosperma females aren't invaded nearly as much, with only 25%. So then we compared this aggressive small sperm species to this very invasive macrosperma that has big sperm to see what happens here. And macrosperma, as we already know, invades in some of its cases, even in some females. Caenorhabditis doesn't quite do the same thing. And then Caenorhabditis doesn't really manage to invade the macrosperma females, but the macrosperma females invade this aggressive small sperm species with 100% success. So I guess in summary, I can say big sperm is more harmful to small sperm females, while big sperm females are more resistant to foreign sperm. So does size matter? For now, the answer is yes, but I'm not done. I'm going to look at more species pairs with big and small sperm. All the ones in blue have big sperm. There's 3 more in the genus that haven't been part of this analysis, but there's a total of 7 out of 50 Caenorhabditis that have giant sperm that I want to look at. I've also started to score some mortality data, so within 24 hours the sperm can actually kill the females. And I want to study the mechanism of invasion, so what's really happening on a cellular level, as well as potentially in the future some ecological implications through reproductive interference and the likes. And with that, I want to thank a whole bunch of people in my lab, my department. They're all really amazing. 2 of them are here. Thanks a lot. Yes.
You said they occur sympatrically and beyond, I think you said.
They do.
Do heterospecific matings ever occur in nature, do you know? Is there any information on that at all?
They're really hard to study in nature because they're really neat along and transparent, so you can't even detect them on their natural habitats. You need a microscope to be able to see them. So it's really, really hard to study that in the actual field. I would love to if it was possible. For all we know is if you throw them together, and even if the right and the wrong male and female are present, sometimes they just mate with the wrong ones. But there might be variation in mate recognition across the different species of the genus. Behavior is just really hard to study in Caenorhabditis. Any more questions? Or if not, that's fine too. Okay, thanks everybody.